Good evening. Thank you, David, so much for this generous introduction and for organizing all this. I'm truly honored to be here tonight, and I'm really happy to have the chance to interact with all of you. So I think David gave a explanation that is better than any that I could have made about my own work, but I'll give you a couple of little more details. So our story starts with this little creature. It's essentially a normal-looking mouse, a mouse that may look like any other you may have seen over the years in your garden, in your basement. But there is something interesting about this little animal. In this particular strain, there is a mutation in its genome. There is a piece of DNA missing in the genome of this particular strain of animal, which otherwise makes it look completely normal, but it has a very interesting deficiency. This animal cannot sense sweetness. It cannot detect the taste of sweet. So if you give this animal sugar or you dilute table sugar in water and give it to drink, unlike almost every other animal in the world, they will not notice anything special about sugar. Neither can it detect the sweetness in the type of chemicals that we use to call artificial sweeteners. So it's a very interesting animal. It eats food, it gains weight, and it carries on with its life, except that it never had this experience of sensing sweet. So just to show you a quick diagram of what this animal looks like, on its tongue, there are sitting there these onion-shaped cells that we call taste cells, which are cells that exist on our tongues and almost every animal that can detect taste. These cells are sitting there on the tongue. They are innervated by nerves that go to the brain. So once a taste is detected by these cells, an electric pulse is sent through these nerves that connect the tongue to the brain, allowing our brain to process this information and create the sensations of sweet, bitter, etc. So this particular animal lacks this gene called TRPM5. We don't need to worry about the name, but it basically lacks a gene on its taste cells of the tongue that prevents it from tasting sweet. So then, many years ago, for reasons that I still don't know, I decided to run the following experiment. I decided to try to understand whether this sweet blind animal could still like sugar even if the sense of sweetness was absent. So I basically, at the time, offered 2 bottles of solutions to the animals, either say water and sugar, or sweetener and sugar, sweetener and water, etc., and try to observe its behavior. So, as expected, in the beginning, the animal was very confused. Everything tasted the same to this animal. But after waiting for some time, something very interesting happened. These animals, they started drinking only from the sugary water. They ignored everything else, and they consumed amounts of sugar that was equivalent to the amounts consumed by the animals that had a normal perception of sweetness. So even without being capable of detecting sweetness, these animals showed a very strong attraction to sugar. And in addition to that, when the brain contents of these animals were analyzed via a number of techniques, the amounts of a neurochemical known as dopamine, these amounts were basically also equivalent to the amounts found in normal animals after they consumed sugar. Dopamine is a chemical produced by the brain of almost every animal, including insects like the fruit fly, that is absolutely necessary for us to derive pleasure and motivation out of our experiences. And it's intrinsically related to our ability to generate motivated movement, motivated responses, like grasping objects and walking towards a certain place in space. So even though these animals lacked the ability to tell via their tongue that sugar is sugar, there was something in their bodies telling the brain that what they consumed had energy, was good for their organism, and this action was supposed to be repeated whenever possible because the animals were gaining in nutrition and energy. So, as I mentioned, the reward system of these animals were activated by sugar even in the absence of taste. So what is the reward system? The reward system is a complex network of neurons in the brain that is also highly conserved across species that, as I mentioned, is the mediator of our ability to derive pleasure, motivation, and motivated movements throughout our life. So here is a side view of a human brain, and this is the back of the brain is here. And at the very bottom of the brain, in an area called the brainstem, we have some of these cells that are situated very deep in the brain and release this chemical dopamine throughout several areas of the brain, leading to our ability to take decisions based on rewards that we acquired in the past. This neurochemical dopamine is essential, as I said, for pleasure, reward, learning, motivated behavior, and this is the neurotransmitter that is impacted in Parkinson's disease, is the primary target of the disease in the brain of Parkinsonian patients. So we were investigating the ability of these nutrient-derived signals from the gut to reach the very centers of the brain that mediate essentially all behaviors that we call rewarding and pleasurable. And this goes all the way from food, from quenching thirst, to form attachments such as romantic love or parental attachment. So the question was how exactly the gut and the nutrients in our gut can tell these reward systems in the brain what is the content of what we just ingested. So it turns out that there is a particular nerve that connects our gut to our brain, known as the vagus nerve, that is essential for this communication, very much unlike what we always believed to be the role of this nerve in animals and people. So the cranial nerve is essentially the longest nerve we have in our bodies. It's also very conserved across vertebrates, and it carries information that it detects in several organs, in particular the gut, but also other organs like the heart and the lungs, and they constantly feed the brain with information about what is taking place in our body, what is our current physiological state. And then we found that this nerve, especially the branches that connect the gut, end up via a relatively complicated pathway that we don't need to worry too much about. They go and connect the gut sensors for nutrients and hormones to the very cells in the brain that release the chemical dopamine that is very important for pleasure and motivated behavior. As a side note, you can see in this list of brain areas the name substantia nigra compacta. And these are the exact cells in the brain that degenerate in Parkinson's disease. For many years, it has been somewhat poorly understood why Parkinsonian patients almost often or almost always present with gastrointestinal symptoms. And the theory was that maybe that the pathology may start in organs such as the gut and is spread into the central nervous system. And pathways like this, the ability to map the wiring connections from the gut to the brain may give us an opportunity to intervene in this type of progressive diseases. Very interestingly, there is a study from Scandinavian showing that patients who had the vagus nerve cut for treating serious cases of ulcers in the past, over the years, they were significantly less likely to develop Parkinson's disease compared to a control group of patients. They have these very large databases. So essentially, this is a frontier area for future research, how exactly our visceral organs may be involved and contributing to the development of certain neurodegenerative diseases by hijacking these nerve pathways that connect the gastrointestinal tract, for example, possibly the lung also, to important centers of the brain that produce the chemicals that we need to behave properly, including produce purposeful motor actions. So the vagus nerve is a nerve that has been studied for centuries. This is a 19th century depiction of the internal nerves of the body, but its function has been always mysterious. And thanks to the new tools developed in neuroscience that have been emerging over the last few years, we now have a better handle of what exactly these nerves may be doing to our body and our brain, because we have a much higher level of control of their activity compared to what it was, say, 10 or 15 years ago. Another interesting aspect of the nerve is that the stimulation of the vagus nerve is one of the FDA-approved therapies for drug-resistant depression. So patients who tend to not respond well to depression medications and who are on the more serious side, they have been recommended vagal stimulation because it was noticed somewhat by chance that their mood ratings improve significantly when they have a stimulator implanted on their neck to stimulate the nerve. We believe that one of the reasons why this may work on a certain group of patients is the fact that the vagus nerve is engaging the reward system by sending these electrical pulses to the dopaminergic cells of the brain. So now there is the development of more targeted, more precise therapies for depression by stimulating the vagus nerve that focus more on the gastrointestinal branches of the nerve. The nerve also has been used for many, many years as a potential treatment for epilepsy, especially in children. And this was somewhat also discovered a little bit by chance in the 19th century. And now, again, there is this idea that this type of therapy may work because the stimulation of the nerve is actually tapping into very important chemical centers in the brain. And this is one way we can intervene on brain physiology without having to operate necessarily the brain directly, but by intervening from peripheral centers. Another aspect of the nerve that I would like to highlight, that David also referred to, is the fact that this nerve does not work only in one direction. It is reading information from the body, but it can also send information back to the body depending on certain types of brain activity. So the vagus nerve can pick up signals from the brain and transmit these signals to a number of organs in the periphery that happen to be innervated by some of its branches, including glandular cells in the gut or the pancreas that release very important chemicals that are very relevant for our health. So one other very interesting function of the vagus nerve that hadn't been really appreciated before is the fact that emotional centers in the brain, for example, the so-called fear center, the amygdala, here this word there, CA, means the central amygdala is one part of the amygdala. They happen, we found, to be direct controllers of the neurons in the brainstem connected to the vagus nerve. So when emotional states arise, negative emotional states take place in the brain and are generated by the brain, for example, chronic stress, or the loss of a loved one, or episodes of bullying or harassment, the amygdala changes its activity very dramatically and passes on this abnormal activity pattern to the vagus nerve. And the consequence of that is that the activity of organs distant from the brain that depend on the vagus nerve will also change. In particular, we know that in positive mood states, in positive psychological states, we, under the correct diet, tend to maintain a normal microbiome in the gut, a normal composition of microbes that enormously help us to digest food and to protect our gut from the invasion of pathogens that come from food, from the environment. So the existence of a normal microbiome containing, for example, lactobacilli and bifidobacteria is very positive and important for our health. However, in negative psychological states, we have an abnormal activity of the vagus nerve caused by the altered activity of the emotional centers of the brain. And this eventually leads to abnormal gut physiology and an inability of these important microbes to survive in the gut environment. So this is one mechanistic way to understand what is the relationship between emotional distress and its impact on physiology, immunity, and gastrointestinal problems. One a mechanistic model for this connection is probably the inability of the vagus nerve to translate a normal brain state into a normal physiological state in distant organs, especially the gastrointestinal tract. Finally, I would point to another, let us say, frontier area of research in gut-brain communication, which is the emergence, the widespread use of weight loss drugs. As you know, there has been a new generation of drugs that act on the brain in order to reduce appetite and benefit patients in a number of ways. And these drugs, especially the new drugs such as Ozempic and Wegovy that you all must have heard about, they basically mimic some of the gut signals that are intrinsic to our body. Right now, the current model is that these drugs act directly on the brain. They change certain brain centers in a way that remove our appetite. However, there are a number of puzzling facts related to the side effects of these drugs that are difficult to explain by just assuming an action on the brain. So another factor that seems to have been not very well appreciated is that these weight loss drugs, they tend to change the state of our stomach, of our gastric pouch, in a rather dramatic way. And this transformation, these changes in gut movement and gut morphology are picked up by nerves, especially the spinal cord nerves, that then send this information to brain areas that reduce appetite. So another important direction in research is try to understand how exactly our brains detect the sensation of fullness and satiety, not by direct actions of drugs on the brain, but by picking up signals that are being generated deep inside our bodies. So to conclude, I would say that the new challenge for scientists working in this field is try to understand how our organs inside our body generate very clear sensations that we ascribe sometimes to our own willpower or motivation. So changes in the physiology and even the morphology of internal organs, they change our psychological life in a way that is not completely conscious to our brain. So we have these signals, and these signals travel in a way that are not directly detected by our brains. So sensations such as hunger and satiety and fullness and nausea are not very well understood yet, and that's why there is so much interest in developing drugs to treat abnormal sensations associated with these states. So we hope to motivate young scientists to tackle this type of issues in the future by using the modern tools and the techniques that we have been trying to develop over the years. So if you would like to know more, I am associated with the Max Planck Institute for Biological Cybernetics. We have this body-brain cybernetics department in the institute that is dedicated to better understand the communication between the body and the brain, how states like hunger, appetite are formed in the brain, how Parkinson's disease is connected to gut pathophysiology and other topics. And I would like to thank you again for the opportunity to speak to you.